Hello everyone. Today I'm going to talk about microarray data analysis. This is the last topic related to your final exam, and lectures 13 and 14 won't be included in your final exam, but they have very interesting topics related to advanced bioinformatics. So I invite you for sure to watch those videos if you won't attend the classes, because they can be very useful, especially if you are interested to do research in the field of bioinformatics. Now, what are microarray data? The data can be obtained using gene chip analysis to measure gene expression. What do we mean by expression? Gene expression. It means that how much a protein can be synthesized using a specific gene. Like, for example, if we are talking about beta globin gene, how much or in how what we can say is that in what level of mRNA can we have, for example, beta globin mRNA or beta globin chain as proteins. So these are related to gene expression. If gene expression is upregulated, it means that we will have higher level of that specific mRNA or protein. So in order to measure that, there are biologists using a specific chip, and by using that, they can gather such data. So this is something like physically available, like a chip, like the ones that you use in computer. This is pretty much the same, and they are using in their laboratories in order to measure them. For us as computer scientists, obviously, it's not that much interesting to know about the technical part of that, but anyway, we should know that how they measure that. Now, the data such as levels of mRNAs and proteins, we call them microarray data. And because one way to find them is using this technique that I mentioned. There are other techniques that I will also talk about them. Let me remind you, how do we get mRNA? By transcribing genes, right? How do we get protein? By translating mRNA. So there's a path between them, if you remember. So if we upregulate gene expression, it means that we will have more protein at the end. If we downregulate it, we will have less. So naturally, this upregulation and downregulation happens in an organism. But assume that we design a drug or we apply gene therapy. So in that case, we are middling with nature and we try to upregulate and downregulate a specific gene expressions. And that's how we can treat maybe disorders or diseases. So this is very interesting topic, and we will talk about it also during the next lectures. Now, the thing is that huge amount of microarray data were collected since last few decades, and because of that, we have lots of data, and we don't know, for example, I mean, biologists, they cannot handle all of them. So because of that, computer scientists and statisticians, mathematicians, they are very useful in order to help biologists to deal with that. And biologists, of course, they understood that they need to learn statistics more than ever, so that we can deal with this huge amount of microarray data. So we have a big number of research committees which are working on this. And now that we have huge amount of data, this is a challenge, but at the same time, this is a good thing, because if you have more data, if you have the knowledge in order to obtain information from this data, then maybe we can cure many diseases, for example, or have so many discoveries. So another method in order to find the level of mRNA, for example, is PCR. And you heard about PCR, especially after Corona, a lot about PCR tests, for example, so that they are measuring the amount of mRNA, a specific mRNA, for example, in our body or protein. And when we say qPCR, we are talking about the quantitative PCR. And in that case, quantitatively, we are measuring them, which is very useful. After that, we can analyze those data and come up with some results at the end of that. As I mentioned, the goal at the end, after having so many data, is to come up with the systems biology. I will define everything regarding this next week. But the thing is that assume that you have, for example, this specific mRNA, and you have another mRNA, for example, here. Now we are thinking that if whenever we upregulate this specific mRNA, is this mRNA in another location upregulated at the same time or not? If it is, maybe there is a relation between them. Not 100%, but maybe there is a relation between them. So in systems biology, we are interested in these things. So if we discover such relations, then we can say, for example, if I downregulate a specific mRNA, maybe I will increase or decrease a specific protein production. And based on that, I can cure a disease or make things maybe better in that organism. So that's why it's a very interesting topic, and we will talk about it even in more details next week. But before going through that, today we are going to concentrate on microarray data analysis methods, especially clustering ones. So there are so many methods in order to analyze this data, but clustering is very common. And you already know about UPGMA. I will talk about k-means briefly. And we have also other clustering methods as well. Now, what we are doing in this clustering method is to grouping our genes into the different categories, into different groups, so that as soon as we talk about putting items inside groups, we remember clustering. This is not always the best method for grouping items for so many reasons. In sampling methods of a statistic, we talk about it. Maybe a stratified method is better than clustering, and there are so many reasons for that we don't talk about it. But for microarray data analysis, these algorithms are very useful, and you already know about phylogenetic trees and how we dealt with grouping species using UPGMA approach. So we are going to practice with this one as well, and also in the weekly assignment, because you're already familiar with that, and we will have better understanding of this one. But also I will mention about the differences between, for example, these two common used clustering methods as well. Now, assume that you have such a table. You have 5 different genes, and we have run different experiments on them. Maybe each experiment was done in different laboratories. Different people maybe worked on it, different research groups, but we have such data. So it continues. I didn't mention, for example, how many experiments we have. I didn't mention any data here, but it means that they have some data here when I put 3 dots. I just want to show you what it looks like, and I will also give you an example. So the question is, which genes should be in the same group? Now, we want to see which method we can use that in order to group them. But first, when you have such a data that you have different numbers, different values for each one of the experiments for the same gene, you see, for the same gene you have different values. This is actually tricky because you expect, for example, if you are measuring a specific gene expression, these numbers, for example, could be mRNA level, they could be protein level. We talk about them like gene expressions, okay? So they could be either mRNA or protein level usually. So we expect that they should be the same, like why it happens like in different experiments we reach different levels for the same gene expression. So in that sense, the thing is that for biological systems, this is totally natural. I mean, whenever you are running experiments, even in the same lab, you receive different values for that. And in 2 weeks later, I will explain you how you can deal with such various numbers or various values that you receive for the same gene. But anyway, the question is how we can group them first. So for this week, we are focusing on that. So in order to do that, you remember for phylogenetic trees, I gave you a score of aligning, and I told you how we can come up with the distance metrics. We can do the same exactly with this one. Now we have experiments, we have different genes. Now I'm interested to come up with the distance matrices for my genes. Genes only. I don't care about experiments anymore inside that table. Okay? So there should be some methods in order to deal with that. I need some distance measure. For score of alignment, I told you about inversing, for example, your scores. Here we cannot do that. Here we should apply another method because we don't have only one experiment anymore. And I didn't give you information about, for example, score of gene 1 and gene 2. You don't have such an information here, right? You have information of gene 1, different experiments. Gene 2, different experiments. We don't know what's going on between gene 1 and gene 2, right? So there should be a measurement. So in order to come up with your distance metrics, you can use different distance measure. So one of them is Euclidean distance measure. And this one is very similar to the one I explained for you for coordination of atoms, right? This is it has the same logic. That's why that one is also called Euclidean method. So in order to find the distance between two specific genes, and you can see that they are the same if you change their position, the distance is the same. Like also if you have 2 points, and this is A, this is B, this is the distance between them. So obviously the distance between A to B is exactly the same as distance between B to A. Okay, now here in order to find the final answer, as I said, this is familiar to you now. You are finding the difference between two genes in a specific experiment. So I here stands for the experiments, okay? Experiment number 1, experiment number 2, and so on. And you power it by 2, each one of them, find the summation, and find the square root of that. It means that here I will subtract gene 1 from gene 2 in experiment 1, power it by 2, plus the difference between these two, power it by 2, plus. I continue until the end and get the square root of that, and you say this is the distance between gene 1 and gene 2. Then you can do for the rest of them and finally come up with your distance matrix. So at the end, what you should have is something like this. Assume that this is gene 1, gene 2, and so on. Again, you will have gene 1, gene 2, and so on here. Okay? So at the end, you should have such a table, such a matrix in order to finally be able to group them. How can we group them? Then it depends on the algorithm that I would talk about it. But first thing first, we should know how to find the distance matrix, which is important. Now, what's the difference between G1 and G1? As I said, for distance matrices, always consider 0 for the same items, okay? So distance between G1 is always 0, okay? We should be careful about that. In the inverse of a scoring matrix, we did the same. We didn't care about the value. For example, if it was 7 as the score of A and B, and if I ask you what's the distance between, A and A, both of them are the same. Now, if I ask you what's the distance between A and A, then this is 0. You don't get the inverse of that, okay? So we should be careful about the diagonal of the distance matrix, which is always 0. And for the rest of them, it depends in which method we are using in order to find the values of this one and so on. So as I said, if you find this one should be exactly the same, right? So you need to only fill this part of your distance matrix. There are other measurements for finding the distance matrix. One of them is cosine. For cosine measurement, what we are doing is to find the summation of the multiplications. And for this part, you should power them by 2, each one of them separately, and get the summation of them and find the square root. So there are different measurements, as you can see. Like for this formula, it means that here, what I do is to multiply these two values together, and find the summation of them. This is related to this part. And for each one of them, you just power them by 2 and add them together. For this part also, you should do the same. We will practice them together. And for Manhattan distance measure, this one is also very famous, very similar to the one that we are using for Euclidean. But in Euclidean, we are powering by 2 and at the end get the square root of that. Here, without doing these two operations, we just get the absolute value of the difference and get the summation of them. This Manhattan's distance measurement also very famous. It means that you just find the difference between each one of them, and if this is negative value, make it positive. That's it. And you get the summation, and that's all. Back in statistics, we have Pearson correlation for comparing two genes together to find out whether they are related to each other or not. This one is very famous. We are using it a lot in order to find about the relation between genes. This one I won't ask in the final exam because we are practicing with this in the statistic course. So I'm not going to talk about it in more details, but what you should know is that by using such correlations, we understand whether two genes are related. For example, if gene 1 is expressed in high level, or better to say the expression of this gene is upregulated I'm interested to see how much does it affect my second gene. Is it upregulated too or downregulated, and how much? So by using such correlations, we get idea whether they are related or not, and we can discuss about it accordingly. Now, as soon as you find out about your distance metrics, the next thing to do is to use an algorithm in order to group your genes. So we have several genes. We are interested to see, for example, gene 1 belongs to which group. You remember about this algorithm which we used in phylogenetic, and we will practice with this one again and again so that there won't be any problem, at least in one algorithm. But I want to also talk about the difference between them. What did we do in UPGMA? Assume that you have 5 genes. So gene 1 to gene 5. And what we did here was to find the minimum distance between 2 genes and group them first. Like, for example, I put G1 and G3 in the same group because the distance between these two were the minimum value, right? Or, for example, I found out that G2 and G4 are in the minimum. They have the minimum, so they will be in the same group. And then I understand, for example, G1 and G2 are having the minimum value inside my distance matrix, and at the end I should connect these two and find out my tree. But as you know, for finding the tree and constructing it as a phylogenetic tree, I showed you how we can deal with that easily. This one gives you an idea, but the best way in order to show it, it's starting from the root and show it in better appearance. Okay, something like this, then you continue and you can show me here graph something like this, for example. Okay, this one, by the way, is not related to this. I just wanted to show you the final appearance for your phylogenetic tree. But for k-means, what's the difference between this algorithm and UPGMA? What we do here is to, first of all, determine about the value of k. How many groups do you want to have? For UPGMA, we don't talk about it. We don't know how many groups we have. We just find out the phylogenetic trees for them, and based on that, you can discuss, for example, which gene is closer to another gene, which I prefer also this one because of this. But for k-means, you are forced to have groups, like this is cluster 1, cluster 2, cluster 3. It says, for example, you should have 3 clusters if k is equal to 3. You can, of course, change it based on the question. So assume that, for example, by random, at the beginning of the algorithm, randomly, we are grouping our genes. Randomly it happens without even knowing my distance metrics, okay? But then after that, you construct your distance metrics and then find out what's the average between them. That's why we call them k-means. In a statistic, when we talk about mean, it means that the average of values, right? So I find the average of these two, we call it centroid, and C1, for example, or C12, because this is related to gene 1 and gene 2. You can call them like that. Then this one is C3, this one is C4, 5. Then you compare these 3 together, okay? Now we have distance between each one of them. Now assume that inside this cluster, I have G4. Then I compare G4 with these different values of the means. Which one is G4 is the closest one? So I may find out that G4 was not in the correct group. Maybe G4 should be inside this cluster. Okay, so I take G4 from this cluster and put it here. What does it mean? It means that now G5 inside this cluster will have a different new centroid, and you should find the new mean for that. Again, for here, I should find the new mean for that. And again and again, you update this until you find the best group. At the end, you will come up with something like, let me show you here. This is not related to this slide, I just want to show you here. So at the end, maybe you find out that G1 and G5 should be in this group, G3 should be here, and G2 and G4 are here. How did you understand this is the case? Because when you find out about the means of them, like centroid 1, 5, centroid 3, centroid 2, 4, when one by one you compared them with these centroids, you understood that this is the best group for this because you reached a minimum value for that. So that's how k-means algorithm works. But I don't ask you a question about k-means algorithm in the final exam because you're already familiar with UPGMA, so I want to concentrate on this one. But I wanted to let you know that there are so many clustering methods, and not just clustering methods, there are so many algorithms in order to deal with that. So in this course, we are just trying to practice with one again and again so that at least you get the idea that what's happening in terms of analyzing microarray data. So as an example, which genes should be in the same group? When we ask such a question, we should specify which distance metrics you should use in order to come up with your final answer. And then we should also tell you which algorithm you use. So you should use UPGMA, and in the question we should specify that as well. And when I talk about distance measurement, so we are talking about these ones like, shall we use Manhattan, shall we use Euclidean, shall we use cosine? So it should be mentioned. Otherwise you can use any of them that you like. So now assume that you have such a table. Again, let's remind ourselves how we can do it from the beginning. We want to group these genes. So we want to use a clustering method in order to group these genes. So the table that you should have is something like this: gene 1, gene 2, gene 3, gene 4, gene 5. Again, here the same for this bar. So if you compare this table with the previous one, in the previous one for the columns you had the experiments, but for the distance metrics you are comparing genes with other genes because that's our goal. We want to group them, and you already know that the diagonal power is always 0 when we talk about the distance metrics. And for each one of them we should calculate it. As I said, for example, this one is equal to this one, so it's good enough to just calculate this one and fill out this part of your table. Now, assume that I want to calculate G1 and G3. So what's the distance between G1 and G3? Then it depends that, oh, we gave you a condition or not. Did we tell you that we need to use Manhattan distance or not? If we don't give you any restrictions, you can use any distance methods that you like. But if we give you restrictions, like to say, you should use Manhattan distance measurement in order to find out your distance metrics. So for that, we should find out the absolute value between each one of them. So I talk about gene 1 and gene 3. So what's the absolute value of the difference between these two plus absolute value between these two, again absolute value of difference between these two, and get the summation of all of them, and finally you will find the value here, which we are going to practice in the weekly assignment. Okay guys, if you have any questions, please let me know. Otherwise, we will practice it in the weekly assignment, and I will talk about it in details, that we should be careful about which part of the table and so on. And next week we will start talking about biological systems and how to analyze them. If you have any questions, please let me know. And I'm going to check your messages now.